diabetes and type 2 diabetes and obesity. We've now changed that a little bit and have coined the phrase the DOHAD, which is Developmental Origins of Health and Disease. And Professor Hansen's going to talk quite a bit about that later this morning. So the marker hypothesis was really built on this database here of a nurse midwife, this woman here, who collected all of this data. This is actually a very nice database compared to what we have in the United States, although it's getting a little bit better. And so from that data that was collected about birth size, a little bit about mom's phenotype and how mom did during pregnancy, he and Nick Hales made the observation that those, again, infants, very similar to what was shown in the Dutch famine, that the odds ratio for the development of diabetes or glucose intolerance in men who were 64 years of age, and again, this is in the UK, in the United Kingdom, and found that those who had a low birth weight of 5.5 pounds or less had a very high rate or odds ratio of almost 7-fold higher risk of developing type 2 diabetes or glucose intolerance. And so now when we think about growth retardation, so for those of you who are neonatologists and obstetricians in the audience, this is a very familiar growth curve shown up here. I think my pointer is dying a little bit, but that's okay. So the top graph shows a growth chart during gestation, and meant to be at the 50th percentile at the beginning of the 24th week, 25th week, and that growth slows. And there's a variety of causes for that. Fetal malnutrition is one. Uteroplacental insufficiency is another. That is a common complication of pregnancy and is associated with malnutrition because there's a decreased availability of substrates, leading to the birth of a growth retarded or SGA infant shown on the bottom, so that thin infant. And so twin studies can sort of help us try to understand these mechanisms, because a lot of these studies were, again, epidemiology studies. There's a lot of data that are missing from those kinds of studies. And so some nice work was done in Denmark and looked at monozygotic twins that were discordant for diabetes, and found that their weights were different, 83 versus 72, which is not surprising as type 2 diabetes does track, obviously, with higher weights. And that the waist-hip ratio, which is a good measure of fatness, abdominal fatness. But what was very interesting is that those twins, those monozygotic twins who developed diabetes, had a much lower birth weight. And they did try to correct for many of the factors that can or are associated with the development of type 2 diabetes. So even after correcting for some of those factors, they were able to make that observation that birth weight was really the single most important indicator or risk factor for the development of type 2 diabetes in this twin cohort. There was a much bigger study that was done in the United States called the Nurses' Health Study, and that is a study of about 70,000 married female nurses. It's mostly a questionnaire-based study. There were some phenotypic measurements done, but most of it is a questionnaire. And that they found then, after, again, trying to correct for all of the factors, which you can't correct for all of them, but for most of the factors that lead to the development of type 2 diabetes, such as parental type 2 diabetes, socioeconomic status, education, race, that again, that there was a twofold relative risk for the development of type 2 diabetes if your birth weight was less than 5.5. And again, it's a continuum in terms of birth weights and the risk of type 2 diabetes. In the United States and in Europe, we tend to see sort of a J-shaped curve, where that if you have a large for gestational age baby, you also have a very high risk. These were corrected for maternal diabetes, so none of these individuals' mothers had diabetes in pregnancy. And so just for you males in the audience, you're not without risk either. So they did a very similar study looking at male health professionals, dentists, optometrists, osteopaths, pharmacists, vets. Again, a very similar kind of study was done, questionnaire, with some phenotypic data available, and found a very similar risk, that again, around a twofold increased relative risk for the development of diabetes later in life if you had a lower birth weight of between less than 7 and down to less than 5.5 pounds. So what about the concept of being small and growth retarded at birth, and what about the catch-up growth? So how much does it matter if you're born small and you stay small, or if you're born small and then you later become fat? So a study was done in India, looked at around 500 children who were born full term. Again, that was a questionnaire-based data in the beginning concerning, again, all of the factors that could increase the risk for type 2 diabetes. They got very nice data from that. And they also then did fasting glucose, insulin, lipid, glucose tolerance, HOMA, which is a measure of insulin sensitivity, and then measures of body mass index and composition, and adjusted for sex, age, and body weight. And that they found that for insulin resistance, so HOMA, that, and this is shown here where the red arrow is that if you were born small, but then were bigger when you were a little bit older at age 8, you had the highest risk rate or of insulin resistance when you're 8 years of age. If you're tall, then you also have a higher odds ratio or a higher risk for developing insulin resistance. But if you're heavy, and you were light at birth, but you were heavy at age 8, your odds ratio goes up to 8. So that catch-up growth clearly plays a role in the development of these diseases later in life. So if your mom and dad were short and you were small at birth, but you were tall and heavy at 8, then you had the highest odds ratio. So you had a 10-fold increase in incidence of glucose intolerance and insulin resistance when you were 8 years of age. So if you have already insulin resistance and glucose intolerance, and you're a little fat when you're 8 years old, just imagine what's going to happen to you when you're 16, 25, 45, or even older. So the issue with these sorts of studies is that it's really a lack. We can't follow that individual from the time he was born to the time he develops these diseases. So animal models were developed to try to help us to understand the mechanisms and really to determine whether or not this was a true phenomenon. So there are a number of animal models of growth retardation, and those are what I'm going to talk about. The most common models are models of fetal malnutrition or maternal nutrient deprivation, both protein restriction and total caloric restriction. A lot of very nice work that has been done in the UK primarily looking at those animal models, first with Nick Hales and Sue Ozon, and some of the work that Professor Hansen has also done with his group, as well as maternal steroids. Those of you who are neonatologists and obstetricians in the audience, you know that steroids are very, very important for fetal lung maturation. And however, repeated exposure to that may place that individual at risk. Thank you. I have a new improved one. There we go. And that uterine artery ligation, and that's the model that we use, and that's really to mimic not only fetal malnutrition, but also placental insufficiency that is commonly associated with diseases such as preeclampsia, which you heard a lot about yesterday afternoon, or high blood pressure during pregnancy, placental embolization, and hypoxia. So what's very interesting is that these animal models, all in very different species. Some of these models have been developed in the guinea pig, in the sheep, in the rat, in the mouse. So very different species, very different kinds of models, but they all result in an abnormal phenotype in the beta cell in the pancreas. So suggesting that the beta cell, which we do know plays a fundamental role in the development of type 2 diabetes, plays a very important role in growth retardation. So that abnormal intrauterine milieu places that beta cell at very high risk for later failure. So this is just going to show you briefly about our animal model. This is done in the rat, and it's a bilateral artery uterine ligation model. And so you can see here's the tail, and the mom's head's up there, and this is the uterine horn. So for those of you who haven't seen the inside of a rat, each of these is a fetus, here, connected to the placenta. And we ligate here, so we tie off that uterine artery supply, and that results in about a 50% reduction in uterine blood flow. Again, similar to what we see in the human when there's significant reduction of blood supply to that fetus. We do get a little bit of fetal death, but surprisingly not as much as you would expect. There's a lot of collateral circulation so that those fetuses are not completely impaired. And similar to the human, there is then resulting in fetal malnutrition, is that there's a lower level of glucose and insulin, as well as growth factors and amino acids. The animals are hypoxic and they have impaired mitochondrial function. And we think that this impairment of mitochondrial function plays, again, a very fundamental role in the phenotype. And I'm going to talk just a little bit about that later. We allow them to spontaneously deliver, and then we make sure that there are the same number of pups in each of the litters from the controls as well as the experimental group to make sure that there are no differences in the availability of maternal breast milk from that rat. And then we foster them to a mom that has not undergone surgery, just in case that there's an impairment of blood flow or an impairment of the ability to suckle. About 50% of the animals will develop very mild diabetes. Over the last several years that we've been doing this particular experiment, the phenotype kind of waxes and wanes. And not all of them will develop diabetes, but they all do develop insulin resistance, and they all do develop beta cell failure with very mild increases in glucose. So they're very mildly hyperglycemic. The animals are fat, so again, similar to the human. So they start off small, the IUGR, the small animals are shown here in blue, and these are the controls. So they do become quite rotund as they age. And here's a picture of one of our rats here. And so they have an increase in body fat. The open bars are the visceral fat, so the abdominal fat compartment, which is what most of the fat is in a rat. They don't eat a whole lot more, but they do have increased fat compartment. But they do have a decrease in energy expenditure, which may play, again, a fundamental role in the development of obesity. So they're kind of like couch potatoes. And this is the beta cell phenotype here. And the graph on the left shows insulin secretion. So we take out the islets from the pancreas and we increase the amount of glucose, and then we measure how much insulin is secreted. So you can see that even at day 1 of life, that there's a significant reduction in insulin secretion. This is again similar to what we see in the human, in the fetus, where some very nice studies were done several years ago looking at insulin levels in amniotic fluid as well as in the cord blood, and found that in SGA human babies that there's a significant reduction in insulin secretion. So the beta cells, the top pictures here is a picture of beta cells of an islet. So this is the islet outlined in green. This is the control, this is the IUGR, and you see what looks like a normal islet. So that there's a normal number of beta cells in the IUGR animals here at day 1 of life, but they're not working right. And we think that eventually what happens is that there is a loss of the amount of beta cells able to replicate. So just like the human, in the rat and in the mice, there's a significant increase in beta cell mass over the first 2 weeks of life. And so that increase in mass is due to increase in replication of existing beta cells. And that process is not occurring. So not only are the beta cells not secreting insulin properly, they're also not replicating properly. This is shown here where we've stained for something called BRDU, which measures cell replication. So there's really not much at all here in the IUGR islet compared to the control. And this is associated with a loss of vascularity. So you can imagine that the islet needs to be vascularized because you need to be able to secrete insulin. And so there's a significant reduction in vascularity. And these are all tied together. So a reduction in the ability of the beta cell to replicate is highly dependent on a normal vascular supply. So as the animals age, that insulin secretion and the ability of insulin secretion markedly declines. And this has been controlled for by beta cell mass. And so proliferation continues to decline, which then results eventually, by the time the animals are around 4 to 6 to 7 months of age, in a marked loss of beta cell mass. So in the beginning, we see normal beta cell mass, but as the animals age, because there's an inability for that beta cell to replicate to make more new beta cells, which is the normal process after birth, you see a marked reduction in mass. So here's the islet in the IUGR animal, and you can easily appreciate how different that looks than the islet here in the control. So we wanted to determine what kinds of genes might be dysregulated, and we already knew some of the pathways that were abnormal in the IUGR animal, in particular mitochondrial function, And so we did a survey to try to understand all the different pathways that might help us understand what the phenotype, the complete phenotype was. And we weren't actually very surprised in that we found genes that regulated mitochondrial function, inflammation, oxidative stress, angiogenesis. Obviously, we expected that based on what we had seen in terms of a decreased vascular supply, glucose metabolism, insulin secretion, beta cell neogenesis, making new beta cells, as well as chromatin structure and maintenance, and I'm going to talk a little bit about that. So many, many pathways were altered in beta cells in the IUGR animals, and this was found in the fetuses. So even shortly after that uterine artery ligation, between 24 to 48 hours later, you still find profound changes in expression of lots of different genes. So this is just a summary of lots of work done by many different students and postdoc fellows in my laboratory. And we find that there are changes not only in the beta cell in these animals, but also in the muscle and in the pancreas and in the liver. And what I mean by a hungry muscle is that there's significant impairment of glucose utilization and the ability of glucose to be transported into the cell, leading to insulin resistance. And the same with the liver, and so that their liver is making too much glucose. So those of you who had breakfast, insulin is secreted, insulin comes, talks to the liver and says, no more glucose, you need to stop producing glucose. But that doesn't happen in the IUGR animal. It doesn't happen in the type 2 diabetic in the human. So a very similar phenotype, as well as the burned out pancreas. Now, a number of investigators across the world have made very similar observations, so that our animal model is very similar to many different animal models, suggesting that there are many similar pathways leading up to the development of type 2 diabetes, both in the human as well as in the rodent. And all of these seem to be linked to malnutrition as well as mitochondrial dysfunction. And what we find is that there's impaired mitochondrial function in the pancreas, in the liver, in the muscle, and the brain. And these are just different pathways. So the Krebs cycle is dysregulated in the IUGR rat, in all of these different organs, as well as the electron transport chain is also dysregulated, impairment of production of ATP, which then will result in the formation of free radicals, and with a decrease in the free radical scavenging system, which will eventually lead to oxidative stress. And this has again been shown in a number of animal models. Sue Ozan has also found similar changes in mitochondrial dysfunction, primarily looking at the muscle, and she's also found these similar changes in the human. So which again, I think points out the role of abnormal mitochondrial function in the phenotype, both in the human as well as in the rodent, and suggests that this is a key player in the development of this phenotype. And so now, in the last few minutes, I'm going to talk about epigenetics and try to give you a background and not give you all the little experimental details. And so epigenetics has now made the popular press. So in Time magazine, it's also made the cover of Newsweek. And really, I think, is trying to inform us about the changes that are not only in the genome, but in the epigenome. And so what do I mean by epigenetics? Well, it's a change in phenotype or gene expression that's caused by mechanisms that are other than changes in the underlying DNA sequence. So some of you probably know identical twins, and they look alike pretty much, but they're not always the same, are they? And so epigenetics is one mechanism by which those identical twins, one might have diabetes, one may be fat, one may be rich, one may be poor, and why those differences might occur. And so that these changes may remain through cell divisions for the remainder of the cell's life, and may also last for multiple generations. So this is a very good experimental animal, other than identical twins, to tell us about how epigenetics might work. So these two mice here, the yellow fat one and the nice skinny brown one, are identical in their genes. So they have the identical genetic sequence, their DNA is identical. However, they look very different, don't they? One is fat, and one gets diabetes, and the other one does not. And that's due to changes in epigenetics. And this picture on the right-hand side depicts changes in methylation at this particular area of the DNA in this mouse, in the agouti mouse. And one has changes in methylation and the other doesn't. And that will then result in a very different phenotype. So same genes, very different phenotype. So this really tells us a little bit about what DNA methylation is. So for those of you who are medical students, you may have learned a little bit about this already. And that's the addition of a methyl group to the 5 position of the cytosine. So shown here, up here. So, okay, this is di2, but that's okay. You can see the big M, that's black, so that's the methyl group. And it's catalyzed by enzymes called DNA methyltransferases. And it usually occurs in a CpG context, but not always. So some non-cytosines can also be methylated. And it can be inherited through cell divisions, but not always. And it can alter gene expression, but again, not always. And it's typically removed during zygote formation and then reestablished through successive cell divisions. And it's involved with exon activation and genomic imprinting. And about 60 to 90% of all CpGs are methylated in animals or in mammals. And this is a very nice study that just showed you, again, when I talked about identical twins, that the patterns of DNA methylation change as we age. So the patterns of the 3-year-old twins are pretty similar, shown here on the left-hand side, compared to the patterns of DNA methylation of the 50-year-old twin, which again, it looks quite a bit different. So not only with aging, but even in identical twins, the patterns of DNA methylation in the genome are different between identical twins, suggesting, but not proving, but suggesting that that may be one mechanism by which identical twins are different. The other very well-studied epigenetic change are the histone code. And so your DNA is wrapped around these proteins called histones. And this shown on the bottom here are the histone tails. And so these can be modified, these post-translational modifications. And so the combination of these different molecules can attach to the tails of these proteins, shown here and here. And that can alter the conformation of the DNA wrapped around them. So the DNA can be wrapped around and can compress like beads on a string, like pearls. And it can influence transcription by altering this packaging. So if the DNA is not quite as tightly compressed, you can allow for transcription, whereas it's tightly compressed, it's silent, so transcription is not occurring. And that's occurring by allowing or preventing access to DNA binding proteins. You can also modify nucleosome surface, again, those beads, to, again, enhance or impede recruitment of these effector protein complexes. So the histone code, which I just talked about here, you have a combination of different sorts of post-translational modifications. So when I talked about how there are beads on the string, silent chromatin, which is all compacted, you often see these kinds of post-translational modifications of a specific type that will alter that conformation. Whereas euchromatin or active chromatin that's actively transcribing genes is opened up and is associated with these sorts of translational modifications, post-translational modifications that are active marks. So I'm just going to briefly talk about how we can use that concept of epigenetics to try to help us understand some mechanisms. And I picked one gene here to talk about. So this is one gene that has been noted to be very important in the development of diabetes in the human. And it's expressed mainly in the pancreas. And if you don't have it in the human, you don't get a pancreas. So it's obviously really important. If you only have half of it, so a heterozygous mutation, causes MODY, and that's mature onset diabetes of the young. That's a very common, I wouldn't say very common, but it is the most common form of genetic types of diabetes, and it's actually fairly common here in the Middle East. About 10% of type 2 diabetes is a result of this particular genetic defect. There are many different kinds of genes here that these particular mutations will lead to the development of type 2 diabetes. So expression, so the amount of this gene or the gene product, the RNA, is markedly decreased in our IUGR islets. So shown here in the fetus in the blue, and the 2-week-old animals, 7 and 15 weeks. So you see this progressive decline, 26 weeks, a progressive decline in PDX1 expression. And this is due to changes in epigenetics. So we see silencing marks, these histone modifications, and we see a loss of these active post-translational modifications. And how do we know that these epigenetic changes that we see really result in a change in gene expression? Well, you can do experiments where you can reverse these modifications. And when we do that with a compound called TSA, which actually is being used in humans for cancer treatment, which is thought to be, again, due to epigenetic changes. But you can see that you get a change or an increase back up of PDX1 mRNA levels. So this is prior to treatment. You take islets out and you treat them with this drug. There's decreased levels before you treat them, and then after you treat them with this compound that prevents these epigenetic modifications from occurring, you get a rescue. So these experiments suggest that epigenetic modifications for this particular gene are responsible for its loss of expression. So back to mitochondria, and we see that ATP levels are decreased, and that the electron transport chain, which I had mentioned previously, and these are just graphs showing you the changes in DNA mutations here. And so mitochondrial DNA mutations also occur, shown in this particular over here. So again, marked changes in mitochondrial function, and this is both in our animal model as well as in the human. And this is just to remind myself to talk to you a little bit about how, again, the mitochondrial dysfunction plays a role in these epigenetic modifications. And so what we do is we take out islets from our animals up here. These are islets. They look nice and healthy. We culture them. These are actually islets that have been cultured for 24 hours in a compound that inhibits mitochondrial function. So it inhibits the electron transport chain. We don't kill the islets. They actually look pretty good. But what we see is that there's a marked reduction in PDX1, our favorite gene expression, which is associated with changes in epigenetics. So here's a loss of PDX1 expression shown up here in the blue bar. And this is then, again, you lose the active marks, so you have these histone modifications, some DNA methylation, and gain of these repressive marks. So very similar to what we see in vivo. So epigenetic dysregulation has been shown in a number of now animal models and in the human as well, in all kinds of different tissues, in muscle, pancreas, liver, and fat, by a number of investigators, including Professor Hansen as well as Sue Ozon and Ann Ferguson-Smith. So this has been shown in different animal models as well as in human. And this is just one very nice study that was done showing that there was persistent epigenetic differences associated with the prenatal exposure to the Dutch famine in the human. And this is another study showing that, again, DNA methylation was altered in these humans after exposure to this famine. It was also sex-specific in a number of different genes, primarily in these imprinted genes here. And many of these imprinted genes regulate growth as well as regulate fatness. And one last study that was done actually in humans who had type 2 diabetes showing that the promoter is altered and it's hypermethylated in individuals who have type 2 diabetes, and this particular gene controls mitochondrial dysfunction. So it brings the full circle back in that in the human there are changes in mitochondrial function, in individuals who were growth retarded at birth, and in individuals who later go on to develop type 2 diabetes, and that these are associated with changes in epigenetics. So we think that, again, the mitochondria play a fundamental role, and that epigenetics is involved. There's a lot of work to be done to determine whether or not really that these epigenetic changes that we see are responsible for a phenotype. It may be that they're just a biomarker.
5 minutes, Professor.
Yeah, I'm actually all done. And all of this work could not have been done without the help of many individuals in my lab. In particular for this project, a number of technicians, as well as students, graduate students, and postdoctoral fellows, as well as collaborations at the University of Pennsylvania, the Albert Einstein College of Medicine in New York, as well as funding from the NIH as well. So thank you very much. I guess we'll take questions after the panel.
Yeah. Thank you very much,
thank you.
Professor Rebecca.